Hi, welcome back to our learning channel. Once again, this is Janrux, your host for today. For today's video, I am going to discuss about the monohybrid cross and dihybrid cross under genetic traits and probability. Alright? So, at this point of time, I am going to explain to you what is the difference between monohybrid and dihybrid cross under Mendelian principle. So, there is somehow a big difference because in monohybrid cross we just only use one trait, while in dihybrid cross we use a combination of two traits. How are we going to apply and solve using Pritzker's method and Punnett square? So in this example that I'm going to show you, I will use only Punnett square. Of course, first, by solving those monohybrid cross and dihybrid cross under genetic or Mendelian principles, right? We have to take note always about the traits. So what is the specific traits that we are going to use? And what will be the alleles? Like its dominant or recessive allele? Is it homozygous or heterozygous? So after finding out the traits, alleles, and then the possible gametes by crossing the two traits, round seeds and the wrinkled seeds. So only one trait. So all about seed shape, right? So we are going to cross, say for example, let's presume this is a female gamete, round seeds, then crossed with male gamete, which is a wrinkled seeds. So it represents, right after that, we are going to move to create a Punnett square, right? So this one. Then after that, find out how many genotypes, okay, coming from here, how many faces, and then move to the phenotype, the actual appearance, okay? So what can you tell? How can you observe? Okay, based on the color. So you have to be specific about naming. For example, round, then wrinkled. So wrinkled for what? So we talk about seed shape, all right? So what about the ratio, genotypic ratio? And then the last one, phenotypic ratio, okay? So this is just only a summary about a monohybrid cross. And what about dihybrid cross? Let us remember, in dihybrid cross, there are two traits involved. See, for example, fur color and eye color. So these are traits, okay? The fur and eye, okay? Traits, okay? So there it is. Now, we represent by using G for gray and white. And for the second trait, we use black and red, which stands for letter B, right? So at the same time, because this is it might take longer, okay, by solving this one, so we have to read the given problem, then understand and analyze carefully, and then you move what will be possible gamete. So a possible gamete should be like this, okay? So right after you analyze carefully those given problem, okay, and then after that, create a Punnett square. So there are only 16 squares, blocks in dihybrid cross. Otherwise, in monohybrid cross, there are only 4, okay, 4 squares, because it's only involved only one trait. So 4 squares only. And then after that, you finish in Punnett square, proceed to genotypes. And then after that, naming the phenotypes. So there it is. Then the genotypic ratio, and then the phenotypic ratio. So those are the common differences between monohybrid cross and dihybrid cross. This time, I'm gonna focus about dihybrid cross, okay? So I'm going to read the given problem. A male rabbit with a genotype, okay, GG, homozygous, gray, dominant, and homozygous recessive, red, okay? So there it is. With a female rabbit with a genotype of homozygous recessive, what we call this, white, and heterozygous, okay, BB, which is heterozygous, black. So therefore, make a Punnett square and fill out to determine the phenotypes of the proportion of its offspring. So what shall we gonna do? So this is the way I'm going to present with you right now, step by step. First, we have to find out the traits, right? So in A. So in this section, we need to use a dihybrid cross. So what are the traits in dihybrid cross? Let us remember that there are 2 important traits that was being combined, right? So the traits, which is the color of, or it's a fur color, and eye color. So you have to write it first, particular traits. Next, we need to find out the alleles. So first, we have to learn what are the fur color according to the given problem. So first, we will use G, dominant G, which is gray. Right. Next, we have small g, which is white. So this time, we are going to get the allele of eye color, which is part of the second trait in this dihybrid cross according to the given problem. So we will use capital B, which is dominant. Alright? So it stands for black. And the other one will be small letter b, which is recessive. It stands for red eye. So there it is. So what you have noticed in a fur color or color of that fur of the rabbit, which is gray and white. Then the eye color, which is black and red. So it might take a long process before we come up on that genotypes and the phenotypes. And what are those? Genotypic ratio and also phenotypic ratio right here. Now let's find out what are the possible gametes in this given problem. So before we solve using the Punnett square, let us understand and analyze the given problem. A male rabbit with a genotype of homozygous dominant gray pair with homozygous recessive red. So there it is. So homozygous recessive red. And then in the second trait, we need to find out with a female rabbit. So we are going to cross with a female rabbit with a given genotype with homozygous recessive white. So this one, homozygous recessive white, and pair with heterozygous black. So now, first trait, homozygous gray, homozygous recessive red, homozygous recessive white, and heterozygous black. Right? So here, now we already have the possible gametes. Okay? Now we need to fill out and determine the phenotypes and the genotypes of these of the possible offspring out of these 16 squares. Okay, so let us continue. So first thing, determine part 1 of the traits in this dihybrid cross. So first, pair this one, which is GB. Next one, we have GB. Next, we have GB. And the other one will be GB. So we use a segregation or a law of segregation under Mendelian principle. Next, another trait. We use this one, GB. The next one, GB. Next part, GB. And lastly, we have G and B. So these are applied according to the law of segregation under Mendelian principle. Now, what are we going to do next? So we will do the pairing with the same trait. So right after we form under the process of segregation, right? So first, you have to write this down, and this one. Write this one, and this one. Okay? Write this one, and this one. Write this one, this one. Write this one, and this one. So they have the same appearance or similar traits right now, as you can see. So it's not much really clear, right? So we just try to figure out the correct process or the correct step. Next, we move to another trait. So finally, we are done, okay, by filling out all those blocks or squares. So there are 16 squares. Let us remember that in dihybrid cross, there are 16 squares involved. It is because of the two identical traits which is being combined or being crossed, okay, in this particular process. So in monohybrid cross, we just only have 4 squares, okay, but for dihybrid cross, there are 16 squares. So it might take longer before we come up with the genotypes and genotypic ratio, and including this one, the phenotypes and the phenotypic ratio. Let us remember, guys, that we have to understand between the difference of genotypes and the phenotypes in this process. Right after we finish solving or filling out all those blocks, 16 blocks, okay, of the possible offsprings. So this time, in this process, we are going to determine the genotypes. So what do you mean by the genotypes? So it is a representation of the combined traits. So like for example, we will use this one. Okay, So heterozygous gray and heterozygous black. So this is the first genotype. And later on, we are going to find out how many according to a corresponding ratio, genotypic ratio. Okay, so next, so how many do we have here? This one, the same, you have to count that one, write it out. So make sure those genotypes have or must have the same appearance, same face, same face, same face. Okay, the same, the same, same, same. There it is. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so that was great. So we found out that there are 8 heterozygous gray or hybrid gray and heterozygous or hybrid black. Okay, so there it is. Okay, next part we have to find out this face or appearance. Heterozygous gray and homozygous recessive white. So how many? The same, 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 the same. So therefore there are 8. Okay, so what we have noticed, there are 8. This time we need to find out the genotypes, right? So we are done solving and filling out those 16 blanks on the Punnett square. So we found out on the result that having gray fur and black eyes, there are 8 possible offsprings. While having gray fur and red eyes, there are 8 possible offsprings. Alright? So what about white fur and black eyes? So the result is not here. So there's nothing. So we have to put 0 out of 16. Okay? And what about white fur and red eyes? So the result, okay, try to look at here, but it's nothing. So it's not written here like this face. So therefore we put 0. So take note guys that there are 16 squares only. So therefore, having gray fur and black eyes, out of 16 there are 8. While having gray fur and red eyes, there are only 8. Okay, so these are. Okay, so out of 16. This time we will move to find out the phenotypes. So what is the difference between genotypes and the phenotypes? So in genotypes guys, we just only write letter or letters or a pair of letters, either one trait or 2 combination of traits. So say for example like this one. But remember, alright, so this representation, okay, that depends, it's either homozygous or heterozygous allele, alright? So let's move to the phenotypes. How are we going to write those phenotypes coming from the result on the Punnett square on the possible offspring from the genotypes? So first, we just only write the word. That is the actual representation or whole description and actual appearance. So by writing the phenotypes, for example, okay, gray fur, black eyes. So this is how the way we are going to describe the actual appearance of a certain organism. Okay, so next one we have gray fur and red eyes. So this is the second appearance. The other one we have white fur and black eyes. Okay, there it is. And the last one, so the fourth phenotypes we have white fur and red eyes. Okay, so where do we get this one? In this representation of letters, these phenotypes will be taken coming from the result of the genotype. So means to say that phenotypes, it explain and describe the actual appearance of genotypes. Because in genotypes, we just only use letter representation. Okay? So this time, we will move to genotypic ratio. So how are we going to identify the genotype ratio? So of course, just take a look back on genotypes. So this one, we have 8. The other one, the other offspring, we have 8. This one is 0. And the other one is 0. Or we could have 8 is to 8. This one. Alright? Now, what about the phenotypic ratio or the phenotype ratio? So there it is. How many offsprings got gray fur, black eyes? So let's take a look. This one, this is gray fur and the black eyes. Alright? So there are 8 again. Then how many got gray fur and red eyes? So there it is. So this representation. Okay? So we got 8 possible offspring for this. What about white fur and black eyes? How many? So the result is not here, so therefore it's 0 again. And then what about the fourth phenotypes, white fur and red eyes? Okay, so there are, okay, so try to find it here, it's nothing. So therefore it's 0 again. So the result is 8 is to 8, total of, okay, so 16 offsprings, okay? So in one Punnett square. So this is the way how are we going to come up. But first, it is very important that we are going to understand and analyze carefully about the given problem in dihybrid cross. Why? Because these are involved not only one trait but also 2 traits. So this is one of the most basic application for our students, okay? By teaching them on how to solve monohybrid cross, dihybrid cross, okay, using a Punnett square. Now, there are other ways also that we can use aside from crisscross method, but this is the best and funny ways that we can apply by solving those cases when it comes to genetic unknown probabilities. Alright, so this is the end of my presentation. Thank you so much for your attention.